So first of all, I think I want to personally congratulate Stacy and her colleagues from the Cholangio Foundation. I think, I've known Stacy for quite a while. I really think that, through the persistent efforts over the years, it's nice to see the Cholangio Foundation maturing into the current condition. And also, I want to personally thank my colleagues, Milan and Nabil, for putting together a very vigorous scientific program. I personally have learned quite a lot today. I think, all of us sitting in the audience, very few will argue with me, regardless whether you are a basic scientist, you're a physician, a nurse, a patient advocacy, or you're actually working for a biotech or pharmaceutical industry. I think ultimately what we're doing is really trying to improve the patient outcome so that, we can let these patients suffering from this disease to live longer. And to do that, there's absolutely no shortcut other than running very, very well conducted clinical trials so that we can actually really prove the point that any treatment, any drugs can actually improve the overall survival. And also, we have learned quite a lot with the excitement on the scientific side. I think, how to translate these findings very timely to the bench side, and from the bench side to the clinical side, I think this is really, really very demanding for all of us sitting in the audience. With that, I think, I would like to share with you my personal thoughts on the current challenges of clinical trials in cholangiocarcinoma. But I think, I'm very happy to be joined by Mitesh Borad from Mayo Clinic, and also Eric Collison from UCSF. I think three of us, will try to give you a very brief perspective. Why we think it's challenging, what's the bottleneck that we're facing for the trial design in this area. And Mitesh was specifically discussing a little bit about the logistics of the clinical trial design for the target drugs. But I think, we'll take Eric's expertise, really letting him to outline for us what's the challenge that we have to identify the right signature, what materials that we absolutely need to use, and what's the logistics that we need to consider when we actually design this biomarker-driven study, molecular classification-driven study. So as we heard quite a lot in the last 1 day or so, there are tremendous challenges in the design and execution of clinical trials in cholangiocarcinoma. Some of them are actually very much inherent for this type of cancer. We've been talking about this over years. It is rare, and we have actually tremendous clinical and biological heterogeneity for this type of tumor. We don't have enough support, both from the government or from the institution, and sometimes the coordination of the trials is very challenging, and particularly if you don't live in the UK. So in my opinion, I actually personally think, the major issue perhaps is the last one, and that is, until recently, I think the genetics landscape and also the relevant targets for intrahepatic cholangio and other cholangio subtypes are very poorly defined. And for that reason, we actually do not know what's the relevant target and how do we actually interrogate this one for clinical trial design. And I'll give you an example. I think, we have other rare tumors in oncology, gastrointestinal stromal tumor, acute myelogenous leukemia. These are tumors with definitely lower incidence than even cholangiocarcinoma. So just the inherent difficulty of being rare is not the excuse not to have a successful trial. So I think, over the past several years, fortunately, we're actually seeing quite a lot of momentum. We have actually made a lot of very interesting progresses in terms of identifying the right signature in cholangiocarcinoma, particularly for intrahepatic cholangio. We heard about a lot of exciting talks on the identification of IDH, the fusion FGFR2, and also, the prevalence of these signatures are actually not as rare as we initially thought. Some of them actually could be as common as 20%, and we heard a presentation yesterday that the FGFR2 fusion could be as high as even up to more than 40%. Obviously, that remains to be validated. But I think with this well-characterized marker, it really provides us, with the right molecular technology to design the right study to interrogate if these markers are actually really, really relevant for therapeutic intervention. So where do we stand? I think in the next few years, we are going to see several phase 3 trials maturing globally, and some of them hopefully will change what we do in our clinical practice. So these are the phase 3 trials that, if they actually show the definitive benefit, it can really, really lead to the practicing changing treatment regimen. There are 3 adjuvant trials conducted in Europe, looking at capecitabine, gemcitabine, oxaliplatin, gemcitabine, cisplatin. All these are actually compared, with observation in the resected setting. These trials do include the R1 resection. So I think, if any of these trials really definitively demonstrated, the improved disease-free survival or overall survival, we could actually change the practice paradigm in the adjuvant setting. We're working very hard how to move forward beyond what the ABC02 trial has demonstrated, whether there's any better chemotherapy backbone, or actually whether we can add target agents on top of the GEM-CIS backbone. I think suffice to say, there doesn't seem to be an immediate winner at this point. And also, as discussed in the earlier talk, we are looking at local radiation therapy very critically as a added modality for disease control, and also, whether the disease control can be translated into the improved overall survival. The NRG-GI-001 trial is a very critical trial led by my colleague Ted Hong from Mass General. And also, both Millen and I are serving at the medical chair for this particular trial. So we think it's a very rational trial to prospectively validate, the local regional therapy in this disease. I think the ABC06 trial is a very important trial in my opinion. It would definitely establish the natural history in a very, very prospective fashion in the placebo control with active symptom control, and also, whether full phosphate regimen is definitely going to be improving the outcome for this population. But where do we stand with the molecular target therapy development? I think this slide obviously is a very abbreviated version based on what you heard over the past 1 day or 2. I think we have tested quite a lot of target agents in this disease. Most of them actually failed efforts. Perhaps the most pressing one is what we heard over the past 2 days. Is really try to assess very critically and the IDH inhibitors and also the FGFR2 inhibitors in selected population. So I think, Mitesh is going to go through with us, some of the challenges that we're facing designing the trial. And hopefully at the end of the section, I think as a community, we can perhaps, have a little bit more stronger sense, what we can accomplish. And also, we're actually making progress, as we're speaking. We have actually quite a few well-conducted study, either actually ongoing or actually at the planning phase. The SWOG intergroup trial is really assessing the MEK inhibitor against 5FU-based treatment in the second-line setting. So hopefully this will actually give us a sense whether the target agents is performing better than the chemotherapy. The SWOG 0809 trial is a very interesting study looking at the chemoradiation. I say induction chemo followed by chemoradiation. The study actually quickly finished the accrual. I think the group is actually trying to entertain whether they can move this trial to a large phase 3 trial. And also I mentioned the NRG-GI-001 trial. With that, let me introduce to you Mitesh Borad, who is actually leading the GI group, phase 1 group, at the Arizona Mayo Clinic. So he's going to discuss specifically about the target trial design in cholangiocarcinoma. Mitesh.
Thank you for the kind introduction, Andrew. Also like to thank Milind and Stacy and Nabil for inviting me to the meeting, and it's been a day and a half of lively discussions. Thank you to the audience for sticking around. This is the second to last talk. So I hope to summarize and review really what's been said over the last day and a half, and hopefully also lay out some concrete steps that we can take as a group to execute some of these ideas. Okay, so we know that countries where we are conducting clinical studies or regions where we're conducting clinical studies are actually low incidence regions. So that's why we keep bandying the term rare tumors, et cetera. And this is largely pharma driven in some way, as well as regulatory and clinical trial infrastructure driven. So until we start conducting studies in high incidence regions, there's going to be a challenge on how we do this, and it's not going to be that easy to accomplish at single or even a few institutions if you're looking at low prevalence events. Okay, so a variety of different strategies can be taken. So I'll be spending most of the time going over the targeted therapy strategy here, but we shouldn't forget that there could be a number of different ways to approach the problem, and it may be that still DNA damage and repair is targeted therapy in itself, and how we conduct those studies should also fall into our discussion. With immunotherapy and drug delivery studies, again, maybe sophisticated infrastructures and selecting sort of a few centers and focusing our efforts on doing it through these places may give the sort of highest yield for the effort put in. Okay, so we've had discussions from a lot of stakeholders in the process. Obviously, patients are at the center and focus of all this. But really, to make this work, all these different stakeholders that I listed, and there's probably a few that I forgot to mention, need to be brought to the table when setting these things up, so that there's a common understanding, the negotiations are made up front, and when we proceed with things, we don't run into sort of the stumbling blocks that we could have avoided by not having the relevant folks at the table. Okay, so a lot has been talked about, the genomic landscape of the disease. And, there's a lot of technologies. I don't think I even have all of them here. This is sort of just a sampling of how you can interrogate a tumor. You can do just copy number assays, gene panels, whole exome sequencing, RNA-seq. You can even look at the methylome, ChIP-seq, et cetera. The list is rather long, right? So sort of your interpretation when you do an assay depends on really which test you did and how many of these you did. So it's not a matter of just the truth, but you also have to include the nothing but the truth and the whole truth piece. So depending on what tests you use, the conclusion you arrive at can be quite different. And we would have to think of practical strategies, and Eric will touch upon this in his talk a bit. How to do as much as we can at a reasonable cost within a reasonable time and with the amount of tissue that we can practically acquire. Once we sort of identify, hate using the word actionable targets because even KRAS in some reports shows up as actionable, there's going to be a number of different channels through which physicians try to help patients. Clinical trials would be ideal, but we know that there's lots of other inclusion and exclusion criteria for regulatory reasons and many other patient-specific factors that limit the number of patients that can truly go on a clinical trial. Even in the UK, we heard it was about 7% of patients. We're missing all the other information if we don't capture the data when they go on to one of the other strategies, particularly off-label use. Okay, so again, this is a very simplistic sort of, bucket strategy that's, been described by a number of speakers already. And even the most frequent alterations we've seen are at 20%. The ones we're really going to run into trouble with in terms of execution are the ones that fall 5, 10%, even less in a rare disease. Unless this is done in a coordinated, cohesive manner, those sort of studies will never get done. So the way we're doing trials right now, we heard of two, the FGFR inhibitor study and the IDH inhibitor study. is being done where, patients are assayed for a particular alteration, and only if they have it do they get screened further, and then they go on. So obviously the chance that a patient can enroll on a trial is rather small. And obviously the approach we should take should be the one, again, that's been supported by many prior discussions that we've had, is we really need to take a patient-based approach where we assay them for enough things so that we fall into sort of the 70-80% numbers that Daniela presented in her talk yesterday, so that they can go on to something. They have instead of a 10 to 20% chance, that they have a 70 to 80% chance of qualifying. And obviously this list will continue growing as we keep profiling, keep identifying new targets. I don't think we've really established the complete genomic landscape yet. I think it's still going to be a process in evolution. So, let's take FGFR2 as an example. And let's use the high number of 5,000 being the prevalence estimate. We've seen these fusions so far only in intrahepatic, so we lose a number of patients. Patients may be in a different time course in their disease. They may be getting standard chemotherapy, etc., or they may be too sick. So as you go through all the different things, you may actually end up with a really small number of patients that you can capture, even if you spread the net fairly wide. Now, if you spread it all over the US, you may be able to capture every single patient, which may be about 100 or 150, but otherwise you're talking 70 to 80 patients. And if your study is 50 patients and there's 2 or 3 studies going on, it's going to be challenging practically to get any of those studies done. So we need to keep these kinds of things in mind when doing the studies, and we may really need to use the international model of parallel studies and then pooling the data. Okay, so what are some approaches to increase efficiency? And this relates to the profiling. So obviously the test has to have a quick turnaround. I would say do your profiling early. Don't wait till they have refractory disease. A lot of events are early events. They'll be there even later. Getting analysis once is probably not enough. Doing it at progression, etc., is equally important. Currently, most testing is driven by the oncologist requesting the test. I think really to make this paradigm effective, we need to move into sort of reflex testing where it's driven by the pathologist. And hopefully that will be something that follows. I don't know how many pathologists are here at the meeting. I know there's a few, but again, having their representation here can maybe change that. And then obviously, something that Funda said, don't do a test unless you're going to do something with it, right? So having availability of therapies for the patient in as many regions as possible is important. Okay, so there's other types of trials that can be done just besides just advanced patients to really gauge more understanding and even decide which drugs should be moved forward into more definitive studies. So one would be sort of a window of opportunity trial in patients who are getting resection, because here getting tissue is not going to really be that much of an issue. There'll be plenty of tissue. You can do really all the pharmacodynamic assays you would hope for. You can even test drive sort of the circulating markers. You can get an idea of the PK characteristics of the drug without having to sort of sort all these things out in advanced patients. And as I mentioned, there's different methods through which a patient may get access to a drug other than clinical trials. So if we are to capture this information, we should probably also have a registry-based approach. And I think through collaborations within the foundation, we can create a database and try to capture these key things. Efficacy would be obviously the most important thing we would want to capture, or even lack of efficacy, so that we can focus on the trials we really need to. There's all kinds of different combinations that can come to mind from a hypothesis perspective. So FGFR2 and MAC, or FGFR2 and PI3K, or with the ERB pathways, right? But there's only so many patients and there's only so much time. So if xenografts are generated from at least a few patients with these alterations, you can maybe use sort of these xenografts as a way to keep that patient alive and conduct more of these types of combinations and see which ones, based on this type of data, you want to move forward. So, This is the Cholangiocarcinoma Foundation, but I want to make sure that we don't miss an opportunity when conducting these clinical studies, that we don't leave out the tumors that would otherwise be patients we are seeing as well. And particularly the mixed HCC and cholangio, there's never going to be really studies in that population. So the UK is very well organized, as you saw, or at least that's how it was portrayed. So, we could probably do a similar job that the UK is doing. I think, if you look at these big centers where the population density is, you don't need to open 500 sites to capture everybody. You can probably open a realistic number of sites that fall within these regions so that patients always have access. There'll be, some sort of rural areas or states with low densities where we might have to come up with different strategies. But we should be able to capture 80 to 90% of patients. And these may happen to be where sort of the NCI-designated cancer centers fall. I'm not saying in any way that only NCI-designated cancer centers should participate in this effort, but you can use this as a starting point and then just add a few other interested centers on, and you would really have your network. So, I really think we need to move forward concretely and in the US have a formal research network. This has been done in sarcoma, it's been done in myeloproliferative diseases, GIST, etc. And really that will be the only way to do these trials. This is the Ukrainian parliament. We hope we don't turn out this way when trying to do it. And Lipika talked about some of these aspects that in order to make this successful, we really need to address the difficult conversations up front. And here are 3 of them, and if they're really not addressed, I think efforts, even though there's good intentions, can really fall apart. So, there's a lot of clinical trials now. There's a lot of interest now in cholangiocarcinoma. I hope this continues to grow. And, the spectrum is across a wide range of strategies, not really just targeted therapies. And I'd like to thank patients and their families, funding agencies, and my collaborators for the work I presented. Thank you.
So maybe we will have about 5 to 10 minutes. I know it's running a little bit late with the schedule, not due to our session's fault, but but we're very happy to generate some discussion here. Is Alan still in the audience? Alan?
I'll grab him.
Okay. So Juan or John, maybe you guys can comment a little bit, in UK, what kind of efforts that you guys are making in terms of, centralized efforts to design the next generation of target-based trials, and also, the biomarker-driven study interrogation, yeah.
Thank you. So, as we discussed earlier, one of the big disappointments that we had from ABCO2 is that we were not able to get the funding for tissue collection in the big study. We have now obviously moved towards collecting tissue, and we've done that for all patients in ABCO3. We're doing it in ABCO6, the second-line study, and we will have a very healthy biorepository from the adjuvant study, the BuildCap study, with 421 patients with obviously prospectively annotated outcomes. So at this stage, we're not quite ready yet in order to roll out studies which are testing a specific hypothesis. But the tissue is going to be there, and that is accessible for us to investigate, but also to look at collaboratively. And one of the things that just crossed my mind while we were going through the new generation of studies, which is going to be very much about enriching for what we think may be a correct target and an appropriate therapy, is how we manage patients' expectations. Because I think there's always going to be the romantic idea that we found the panacea that will deal with their cancer. And whilst that might not be necessarily what we mean, that might be what's understood. And I think we need to be very careful of not hyping this too much. I think it is the direction of travel, but I think we still need to have the academic and scientific rigor to be able to test it.
I totally share that sentiment. I personally have a few patients who are actually so desperate trying to get one of these trials, but in the end succumb to the disease before even having any access. Questions from the audience, yeah. Yes, Ranu.
Thanks for a great overview of all of the different efforts. If for some reason you had data right up front before you even started first-line therapy that somebody had an activating EGFR mutation, what would you do? And also, if for some reason there was a GEMSIS or Lotinib study, why are we all still thinking that they need to be given all together as opposed to sequentially? At least in colon cancer, it hasn't really worked that you take all your targeted agents and stick them together.
I'll let Mitesh to take this question.
Well, conceptually, what you're saying is probably, the right way to do it, where if we know that they have an alteration that's highly likely to deliver some benefit, why are we just going the standard therapy? The reason is, we don't know at this stage, so maybe in the initial trials we should study, targeted therapy versus gemcitabine cisplatin as one strategy. And if we find that with a lot of these alterations, we find you should do this up front, then it will become sort of, a more standard approach.
Eric, the cell-free DNA assay, is that as good or actually better than the tumor cell detection, circulating tumor cell detection? Do we seem to bother to chase both technology, or one is definitely better than the other?
Yeah, absolutely one is not definitely better. I think they tell us some different things. There's kind of a, I think of it as a glass ceiling and a glass floor. Tumor biopsying has essentially, theoretically, impossible to tell you about temporal heterogeneity. The technology can't get better to fix that problem. You have to stick with a needle again. Cell-free DNA is inherently dependent on DNA being cell-free. So there are two kind of upper and lower limits that kind of bound the use of the two tests. And I think cell-free certainly has sex appeal now, but at the same time, we don't really know what a negative cell-free DNA test means. It certainly doesn't mean you're not going to die of cancer. It certainly doesn't mean you need good therapy. It just means we can't find anything. So I think there's still going to be definitely a role for both, but not on every patient probably.
Yes, Milan.
Enthusiasm and the hype has sort of gone beyond where we really are. So for instance, we find a lot, so we are trying to do a protocol with you, so I'm a believer it should be researched. But for instance, we have Funda there and I find a BRAF mutation in the blood, will that patient be even eligible for the trial? The current trials are not written in that way. Right. And then often the kind of mutations you find are not necessarily like V600 or whatever, you find something that is not yet described. So it's, I think it's more of a fact-gathering situation right now, and whereas it's being used kind of as a surrogate for the commercial NGS testing in some instances, and I'm not sure that we are close to that at all.
Yeah, I would say that I'm most excited not by the clinical ramifications, but by the clinical research kind of liberation that the technology has. That in pancreas cancer, as you well know, we've just had trial after trial with no tissue, and we never learn anything. And we start from scratch every time and hope this one drug will cure pancreas cancer, and it didn't. And so we need incremental benefits that are not just, I tried to find tissue, but I couldn't find it. And I'm an optimist, and I think that we can actually move forward with technology and learn from our trials, even if they're negative. And without tissue and without other correlates, we can't learn anything from a negative trial.
So Mitesh, I'll let you go ahead. Yeah.
Honestly, I think the data emerging for circulating DNA looks really quite good. And if you look at studies that have looked at concordance of primary versus metastasis or genomic analysis done in one institution versus another, the concordance is not that much better. So I would argue that given the turnaround of circulating free DNA, trials should start considering using this for enrollment criteria and then plan on doing a pre-treatment biopsy, which would be invaluable because then you'll know what you're actually treating. You can do additional RNA versus and protein analysis at the same time and learn more from the patient that has undergone that biopsy.
So Dr. Vinod just walking in. Ellen, we were just talking about, you are probably the most experienced person in the audience affiliated with the cooperative group.
Is that the oldest?
No, I didn't mention age whatsoever, yeah. So in any case, from your experience, when you deal with these kind of target therapy, particularly in a basket trial design, what's the frustrations, even at the cooperative level, that you have much larger access than all of us as individual investigators?
Well, obviously there are too many to count, right? So the first issue is if you've got metastatic disease and you say biopsy it, what do you biopsy, right? So one biopsy, there's heterogeneity enough that you may not be really informing yourself very well. So that's one issue. I think the other issue, there are many other issues, the issues of really having being able to use multiple therapies in a bucket. I mean, the idea of that a monotherapy, single agent is going to target and work is really wishful thinking. Maybe works in a couple of, like lung cancer. So lung is accruing very well, relatively well to their master protocol because there's a sense out there that monotherapy may work. We don't think it's going to work in most cancers, so there's that. And I think the daunting part is the cost involved, the cost of biopsy, the need to turn it around in real time, and then you think of what drugs are available. I think it'd be interesting to see what how Novartis might view this. For example, are they going to take their top agents and make them available in this kind of bucket trial, or are they going to be their second or third string? So I think that's the issue. I think. The match trial is super appealing in the idea if you believe that it's agnostic to the disease, but who here believes that we're really going to be agnostic to the disease? I mean, other than Harold Varmus, I'm not sure many people think that we will be agnostic to the disease. So I think that's it. And then you get to the details of budgeting and all the time, right? How long have we been waiting for match? What is this, 2 years, 2.5 years? So other than that, no frustrations at all.
Very well said. Randy?
Right. So I think I'm saying disease-oriented bucket trials makes a lot more sense to most of us. You guys also have the signature trial, right? Right. The problem there, so there's the frustrations with that, or from our perspective, is the cost involved of doing a separate. It's essentially a different trial for every disease, right? That's how signature is. Every patient is almost a separate trial. So. Which is immensely expensive for the regulatory burden on a site to do that is immensely expensive, right? Because you're replicating all the startup, at least in most places. It's daunting. Right, so that's if you can use quorum, we do not use this. We do not use this. I'm not sure. Some places may use. So in any case, that's an example of an effort being made an extra effort to make it convenient, and yet at least at our place, it's still difficult. Right. Yeah.
So founder, this morning you give a very elegant example how everything is run at MD Anderson for more common tumor types, and also how to use that strategy to develop target therapy. Do you think we can use similar strategy for the cholangio tumor type? And if we do, what mechanism you think will be relatively feasible in the foreseeable future? Yeah.
So I have one idea that we're floating around and we're trying to get traction on is the idea of using the PDX models to sort of choose our winners. It's very complicated, but the idea generally is, in colon cancer, we're talking about doing this across a few centers at least to prove that we can do it. Taking patients at first progression, doing a biopsy, seeding a PDX model, treating them whatever you would with second line, and then informing your third line treatment based on the PDX. Now you of course have to have agents you can put in the model, and then taking the patient before they go on third line, doing another biopsy and making another PDX to see how the disease may have changed under the force of chemotherapy, which has actually been done hardly ever in terms of understanding how the disease changes. Now, what's the yield of PDX, for example, in cholangio? It's very hard to grow, is it not? What's the?
I think we're doing it, and Shoop probably can comment on his stats best.
So primaries are?
Yeah, so we've been taking, these are surgical samples, so this is before.
These resected specimens?
Resected specimens prior to chemotherapy. We've recently gotten a couple of post-chemotherapy specimens, so we'll see. But in my hands, at least, using the right type of immunocompromised mice, I'm getting about a 30% take rate from primary tumors. So I think it's comparable to what other people.
Yeah. Well, it's interesting. So colon, we maybe do a bit better, it seems, but. So, I mean, that's an example of that would be labor intense, but the point is that might give us a better handle on trusting that you actually have agents. But that's a big undertaking. You need, you'd need collaborators galore to do that. Anyway. Right, so Champion Oncology is one that, as you know, we're working with in colon, and I've pitched this to them to look in other diseases, not just the big ticket, not just colon. And I think there may be, that's something we could talk about. I think it's a lot of work, but it may inform us better than everything else we do. What do you think?
I agree. I think this is a disease where we think we have targets, so it's actually optimally poised to do that. But we need to probably do it before the GEMSA starts, because their timeline is it does take about 4 or 5 months to get.
Yeah, 12 to 14 weeks, and it may be longer if it may not. I mean, BRAF mutant colon cancer grows it takes very quickly. The others might not, but that's something we could explore. We're meeting with champions in a couple of weeks to talk. Scott and the MD Anderson and our group are working specifically on trying to do that. So that's an idea. Because but we do it amongst ourselves, and probably you have institutions that can get the tissue, can handle the tissue, and can do it. It does take a lot of moving parts. I mean, the problem is, of course, the model, you can't test immune therapies because they're immuno-incompetent mice. But I mean, so you check those off. But in terms of other things, that would, I think, might be a reasonable way to test it.
Yes or no?
One patient case report to answer your question about growing the tumor. I took care of a chair of one of the departments at Roswell who had had his biliary cancer surgery at Hopkins, and they had a protocol there to grow his tumor. And when he recurred, we got a biopsy. And
He recurred, and he had surgery at Hopkins, and he recurred? Yeah, no.
And both of those tumors grew actually and responded to different chemotherapies. Abraxane happened to be one of them. We also did his testing. One, I don't remember if it was foundation one or which one. And the best response, what was thought to be the best agent that he would respond to was 5-FU and Abraxane, which he got, but the response was stable disease 4 months, and his median survival from diagnosis was 9.5 months, which was not anything stellar. So I just, it's an N of 1, but even when it works, sometimes the results are not.
So, Alan, we've been doing it for pancreas, Jason Fleming and all, Ali Bhan, for about 3 or 4 years, and he is a big farm now with these mice running around with these PDX models.
Lots of cheese.
And I can tell you, honestly, there's been 1 or 2 cases where, like mitomycin worked or something, but they had like DNA repair type of situation, and I wonder if we needed to go through that exercise to find that information.
Well, so I think it seems to me diseases are different, right? That's why we're not agnostic. I think pancreas is a tough nut to crack, and it's like in colon. Why can't we do a sign in colon? Because we can't drug RAS, and that's 60% of the patients have RAS driving it. I agree. I think we have to be bold. If we want to make a difference, I don't think we're going to make a big difference by doing lots of foundation one and getting targets and thinking that a monotherapy is actually going to make a difference. I don't think it's going to get us anywhere.
But it has got us, like Randy and Agios and all that to the table.
No. Well, Randy she wanted to come skiing for the weekend, but no. I agree, and I think the fact this may be more doable than the common diseases. Actually, if you have a group of teams that are actually dedicated to doing it, but I think we have to really think outside the box, because I don't think and I think again we've harmed ourselves with a lot of the hype about the foundation one, the assays, 85% are actionable, and we know that's not the case, but you hear that, and people, and so we are deluding ourselves to think, I think, that monotherapy is going to work, and so.
Mitesh, any additional comments on Alan's enthusiasm?
I didn't say it was enthusiasm, but.
Always good to have, sort of constructive pessimism, right? But I think, just the fact that everybody's here shows that, it's possible. And if sort of some sort of research network where people start testing these ideas is constructed, it'll be better and easier for pharma to want to engage with our sort of research community, it'll be better for patients, etc.
Well, I think the other thing, you can do a lot of research off of this. So if you, for example, like I don't know if you're doing this in the models, if you take the models and then do organoids or do other ways of testing the activity of agents, I think there's lots you can do beyond that could inform us further down the road. So there's a ton of information you can gain. The problem, of course, with what you're doing is if the take rate is 30% for primaries, what's it going to be for? We don't know what it'll be for Mets, whether you'll get enough tissue with metastatic disease. I mean, that's the other, obviously, the elephant in the room. In so many patients, can barely get tissue, let alone enough to do this.
We are working on, we are developing a protocol for biopsy specimens.
Hurry up.
We're also going to probably eventually try autopsy specimens.
What's the cost? What's your cost? What's the cost for you to do a PDX? Do you have any sense?
No, but we can tell you what foundation charges $1,000 per mouse. That foundation.
Champions, yeah.
The other thing is, we've seen some of Champions data, and we've also talked to Anirban and some of our own work. Tumors that are completely unresponsive, like gemcitabine is a tremendous drug in pancreatic cancer. We've seen some of the cholangiocarcinoma data, these random compounds wiped out tumors in that. So I think it's more a research tool that you can, if you have a research question, that you can actually learn a lot. But as a general kind of a huge investment, with, as a blinded fashion, I don't think that it can be that high yield. It's rather targeted research questions.
I think you're probably right, but I think my view is, I think we have to get started doing things differently at least. I think, and again, the spin-off that we can get may be substantial, but and
Maybe preferentially within academia, I think you could bring together to have a PDX program rather than paying a premium for it.
right, Mayo has. I mean, Mayo has a PDX program. We've probably got 6 or 7 programs right around the table here that we could put together, and I don't know. That's.
Luis, you want to comment a little bit about your PDX program?
I think our experience in some ways mirrors, the experiences that have been discussed. It's, I think, challenging to get up and running. We've, I think, made a lot of progress since Mark Trudy came back from MD Anderson, and because Mark spent some time there working on the pancreas PDX program. And I think one of the key things, some of the few things we've learned is that it's really important to go from clamping the artery to the tumor to the mouse in as short a time as possible. So that's been one of the key messages, for example, and one of the things he instructed us on, and the surgeons all know this, but I think the rest of us didn't quite appreciate it, is that a lot of the time they go into the patient, and the first thing they do is they clamp the blood supply, and then 2 hours later they take the tumor out of the mouth, out of the patient. And then it sits in the frozen section lab for another hour or 2, and then they call you. So by the time you see that tumor, it's been 4 hours since it was ischemic. And so one of the key things that he's done at Mayo is really driving to reduce that time, the time from when he actually in the OR clamps that tumor off to when we get in the mouse. And that's made a key difference. In pancreases, he's getting about a 90% take rate. It's less in biliaries, but I don't think it's very far behind. And then we're beginning also, like you are, with the biopsies. And again, with the same thing, trying to arrange things so that the radiologist is getting the biopsy and we have someone standing there waiting to pick it up and put it in the mouse. So I think there's things that we can work on that'll help.
Any other burning questions from the audience?